see why I always love to come here. I mean, this is great. Thank you, Cody. Well, thanks very much for inviting me, and what I'm going to try to talk about today is some of the things that are on the horizon, both near and maybe somewhat near. I'm going to be talking about stem cell technologies, where they stand now and where they're going, and I'll teach you a little bit about stem cells, what they are and what they do. Before I start, let's see, so there we go. I actually am a political appointee now, having been appointed to the committee that actually oversees the funding of Prop 71 in California. So I found more and more that I have to start all of my talks with disclaimers. So who am I? Well, I am the director of the Reeve-Irvine Research Center at UCI, so I actually am a working scientist. Well, my students and postdocs might quarrel about that, but I claim to be anyway. I actually do still do research. I really appreciate the fact that I have been given an MD as part of my attendance here. I actually don't have an MD, but which is everyone is very thankful for. I am a scientist, a PhD scientist, and from a political standpoint, I'm a member of the Independent Citizens Oversight Committee. This is the ICOC that actually oversees Prop 71 funds in the state of California. And I was actually appointed to that committee by Governor Schwarzenegger, and I've learned a great deal from my membership on that committee, including how to spell his name correctly. I have no interest in any company involved in stem cell development, no financial interest, and in fact, I'm disallowed from having any financial interest because I am a part of this oversight committee. So that's who I am. And let me just tell you a little bit first about the University of California, Irvine and the Reeve-Irvine Research Center. We were established back in the 1990s, early 1990s. I was the first director and came on board to chart the course of this new thing. Our mission is to find new treatments for spinal cord injury and other disorders affecting the spinal cord, and we really work collaboratively with people throughout our university system. We want to run a state program for funding spinal cord injury research in the state of California, and I'm also involved in collaborations with scientists throughout the world. I say that because a lot of times people say, well, do you guys actually talk to one another? Do you collaborate? Do you have very many secrets? And the answer is no, we really don't have too many secrets. And really the only competition we have, serious competition, is competition for funding, because funding is so tight right now. And scientists in general truly love to get together and share data. And I guess, I'm standing here talking to you guys in the state of New Mexico, and one of the things that the state agencies are starting to do more and more is fund different kinds of research. And one of the options here is actually to fund people working together, not just individuals. And I would say that we've been able to do that in California to some degree with our spinal cord injury research program, which is actually pretty small. It only funds $1.5 million a year. But still, we work together as a group, and it's really been quite a different situation than the kind of thing that you do when you apply for a grant at NIH, because really fundamentally, NIH will fund individuals or small groups working together at the same institution. But it's very hard to fund projects that are cross institutions. So it's worth knowing for you guys who are out there being politically active. For those of you who are interested in spinal cord injury and what kinds of things are available, we do have a website. It's very simple, www.reeve.uci.edu. If you'll give me 2 seconds, I'll just quickly show you the kind of information that you can find on there. I have to do it from back here, but it won't take long. So basically, if you log on to our website, you'll see something like this. You may not be able to read it, but you have things like newsroom, you have different kinds of programs. You have something called Anatomy 101, so you can just click on that, and here, for example, there's a whole piece on parts of the brain and spinal cord. You can click on stem cells and somatic cell nuclear transfer and learn all about that if you want to. And I will have a handout available before the end of the symposium here with that website on it for those of you who are interested. But it really is pretty easy to remember. So back to the slideshow here. So what I will tell you about is really what we hope to be the future of regenerative medicine. And I guess by way of introduction, everything that you've heard about today involves damage to the nervous system. So damage can occur in a variety of ways. Traumatic injury is the one that we think about most often when we say the words spinal cord injury. Immunological disorders like TM, multiple sclerosis, degenerative diseases like ALS, Lou Gehrig's disease, all cause the loss of particular types of cells in the nervous system. And once that's happened, you can't intervene anymore in the process that made it happen. So we've heard about TM. It's an event, and people either recover or don't. And wonderful things have been done. You've heard about them all that have made incredible impacts on people's outcome following that acute episode of TM. But for those of you who were affected and remain impaired, the promise now lies in looking ahead. What can we do to actually regenerate the connections and the cell types that were damaged in this disease and every other disease that affects the nervous system? And so regenerative medicine is a general field that seeks to either replace lost or damaged cells or promote regrowth of nerve connections and the myelin insulations that makes those nerve connections actually operate correctly. So we are working hard to move this forward, and I'll tell you today about some of the things that are going on in the world, particularly related to spinal cord injury, and that involve this regenerative medicine approach. So I'm going to focus primarily on stem cells, and I'm going to focus primarily on human embryonic stem cells. And of course, there is both science and a lot of politics involved here. And I will tell you things that I believe as a scientist to be true. I will try not to have my personal political opinions color very much of that. And my politics, I think by the end of the day, or even now, probably pretty obvious. But since I was appointed by a Republican governor, I try not to make those politics too obvious to anybody, at least in California. So he knew I was a Democrat, by the way, when he appointed me. So I'd like to just spend a few minutes talking about what human embryonic stem cells are and how they're obtained. So, of course, the word human embryonic stem cells implies that these cells come from human embryos. And I'd just like to ask you, just a little straw poll here, you guys who hear this term, how big do you think an embryo is from which we get human embryonic stem cells? And I'm going to ask you to raise your hands here. So do you think that a human embryo from which we derive stem cells is maybe as big as my fist, maybe as big as an apple? How many think that? How about as big as a bar of soap? How many think that? Okay. How about the size of a dime? How many think that it's the size of a dime?
I'll go for the dime.
Go for the dime, okay. So so we're getting there. So, I'll tell you, the embryos are round, so, so a round thing about the size of a dime. Hands? Any more hands? How many think they're smaller than that? So, yes, those of you who raised your hands that way are correct. And just how small, actually, I think we can see from, again, the dime here. So, the blastocyst, the embryo, from which stem cells are derived, is about as big as the eye on Roosevelt's face of a dime. So any of you who have a dime in your pocket, take it out. Try to see it. I can't with my glasses, but so they're incredibly small. And really what they are, we think embryo, but really these things are just at this stage of development. This is just a couple of days after the sperm and egg have come together. And this structure begins to divide, and it divides about enough times to generate about 100 cells. Okay? And those cells form a ball here that's actually the entire blastocyst, and inside the ball there are a few cells here. And those are human embryonic stem cells. So the way we actually obtain stem cells is to break this ball open. These cells on the outside are called blueprint cells. They're not stem cells, but they are the ones that instruct the stem cells how to grow into the placenta and the embryo, okay? So that when these cells, the stem cells themselves, are taken out, they can no longer develop either into a placenta or an embryo. But what they can develop into is any type of cell in your body. So what scientists have done is to take these cells, extract them, put them in a petri dish here. Ooh, he's a big one. Wow. Yeah, lots bigger. I wish I had had him for a prop when I was actually going through the options here. For those of you on this side, we're talking about a gigantic moth over there, but he seems to have disappeared. So, and these cells are then just grown in little plastic petri dishes. And really, the importance of these things is now that they can be taken out to essentially infinite volumes. We literally are to the point that companies are taking stem cells isolated from embryos and growing them in barrels. And this is what would be necessary for therapeutic applications. And I'll come back to that in a second. So then what? We've got barrels of cells. What characterizes those cells? Two things. They will go on to divide forever, we think, and make more of themselves. That's why we call them stem cells. And they will, when appropriately treated, differentiate, that is, become other cell types of the body. So we sort of know how to grow these cells in vat-like quantities, and we're beginning to learn how to cause these cells to differentiate into the cell types that we need, for example, to treat disorders of the nervous system. And so here, for example, is a situation where stem cells have been treated in such a way that they've differentiated into nerve cells. And this is done by treating these cells with different chemicals, with different growth factors. Doug Kerr spoke briefly about this in his talk, and sometimes it's pretty simple. And it's really interesting that although stem cells can develop into any cell type in the body, they actually kind of prefer to develop into cells of the nervous system, the three main types of cells in the nervous system. That's their default state. So in some respects, it's harder to make a stem cell turn into a heart cell, or a cell of the lung, or a cell that gives rise to regulated insulin secretion, than it is to turn that cell into the type of cell that would be useful for transplantation into the nervous system. We're closer. Okay, so I said that there are politics involved, and of course our issue involving these stem cells is that they do come from very, very young human embryos. They come from blastocysts, and the source of these blastocysts so far is leftover embryos that have been generated in vitro fertilization clinics. So couples come together who can't have children in the normal biological way, and the woman donates an egg cell, the man donates a sperm cell. These are brought together in a dish, and that's called in vitro fertilization, and embryos are created, and they grow up to the size that I showed you. And then some of them, actually one at a time usually, is implanted, hopefully only one at a time, or you have multiple pregnancies, and a child is born. However, more are created than are needed for that purpose. And in fact, generally a process of in vitro fertilization leaves over 15 or 20 of these fertilized embryos, and the couple are done. They have completed their reproductive needs or desires and are finished having children. So what do you do with those embryos? Well, right now they're stored, they're frozen, and the couple gets a bill every month to pay for that storage until they don't want to pay for it anymore. I hit something and it went off. There we go. Never mind.
It's your politics.
It's my politics. Hi. Thanks, Doug. Huh? Woo-hoo! All right. I didn't know I hit a nerve. I thought I just hit a button on the... And when the couple is no longer willing to continue to pay for these things to be stored in a frozen state, then they are destroyed. They are discarded as human biological waste. It's estimated that there are about 450,000 blastocysts stored in vitro fertilization clinics in the United States right now that will eventually be destroyed. So one of the sources of the stem cells that we need are these. And they could be used to derive new lines of human embryonic stem cells instead of being wasted. Now, you will ask, why do we need new lines? Those of you who know the politics know that there are human embryonic stem cell lines out there. In fact, Mr. Bush allowed us to carry out research on human embryonic stem cells for the first time in his decision in 2001. We couldn't even do research, or rather, we couldn't use federal funds for research before that time. And he did allow that research to go forward, but as you know, his decision was to restrict it to the lines that existed at that time. It was thought that there were about 60 or so lines. It turns out there are many fewer, and probably only about 8 or 9. Doug, what's your guess? Numbers?
Less than 10 clues.
Yeah. That are actually usable at all. And some of these have been contaminated with mouse genes because these original cells were grown on a feeder layer of mouse cells. We now know how to grow human cells that don't involve mice feeder layers. So let's say we were able to actually generate a treatment from these cells that are admittedly flawed, but we could generate a treatment. Think about what you know about transplanting other organs, hearts, kidneys, lungs, well, lungs no, but livers, you know. You need to be matched, tissue type matched, before you get a transplant in order to reduce the probability of a rejection. And even then, you have to take drugs for the rest of your life to keep your body from actually rejecting this new transplant. To do the first part, that is to match you with a donor, requires a selection of donors. We don't have a selection of donors. We've got 8 lines of cells, each of which came from a single person, and we all know that there's not sufficient genetic heterogeneity there to come anywhere close to having what we need to develop stem cell therapies for the future. So we need, even just forgetting the fact that these cells are contaminated, not up to standards as far as what we can do with them now, they also don't have the genetic heterogeneity to allow us to go forward. And I think that, at least for me thinking about it, the worst tragedy would be to develop a treatment that would only be appropriate or usable by a very small number of people in the world. And that truly would be a tragedy. So that is the reason we need new stem cell lines. I'm going to take you 2 minutes and just give you the definition of what it is that differentiates a human embryonic stem cell from an adult stem cell. You think an adult stem cell must come from adults, right? No. So adult stem cells, by definition, are stem cells that are derived from stem cells in tissue that also contains differentiated cells. That's a really complicated definition, but basically what it means is a lot of these actually come, thank you, from fetuses. So when you hear the term adult stem cells, it doesn't mean it came from an adult. It means that it could have come from an adult, it could have come from a fetus that was aborted for whatever reason. And in fact, there are companies right now who are developing therapies involving, quote, adult stem cells that actually came from aborted fetuses. And this is legal under Bush's regulations right now. I want to teach you one other thing. I know that you've all heard about therapeutic cloning. This is a big political issue, and I just want to teach you what it actually means. And it might have been Doug. Someone else may have mentioned it, but it's a terrible term. It's an unfortunate term because it evokes the image of cloning a person, and it couldn't be further from the truth. No one wants to clone a person. It's absolutely illegal. And as far as we know, it can't be done. We scientists have cloned a few animals, but the likelihood of actually being able to clone a person, a real person, is very small. So what is therapeutic cloning? And a much better term is somatic cell nuclear transfer. And what it means is that you take a donor egg and you squeeze it a little bit to pop out its nucleus that contains its genetic material. And then you take a cell from an individual that perhaps has a spinal cord injury or some other disorder that requires cell replacement, and you can actually, again, pop out that nucleus, and the cell that you use perhaps is just a skin cell or a cell that you get by a scraping inside your mouth. You take that nucleus then and inject it into this donor egg, and you treat it and cause it to begin to divide, and then it actually grows into, guess what? Our friend the blastocyst. So again, the cell with stem cells in the middle. And the idea is that you can then isolate those stem cells, grow them up, but this time they are actually you. They're made up of your genetic material. And so they would be essentially perfect for use for transplantation into you with whatever disability or disorder that you had that could be treated with a stem cell. Now, I will say that this is, at this point, theoretical. It has been done in animals, but there has not been a successful development of somatic cell nuclear transfer in humans. You may know that there was a claim for that by the Korean group a couple of years ago, and that was shown to be fraud, scientific fraud. They lied. So it has not yet been done, and frankly, we don't know yet whether it can be done. But that's the promise for being able to develop cells that actually have your genetic identity. So just a few questions, and you guys probably all know the answer. I actually shouldn't have put the answers up there. I should have taken a vote. But so, is research involving human embryonic stem cells illegal? Well, I didn't know this until I came here, but the answer is, I guess, yes, in New Mexico. But there's no federal law against it. The federal restriction is that you can't use federal funds. So that's NIH funds, that's NSF funds, National Science Foundation. Can't use Department of Defense funds or any federal dollars unless you're using these stem cells that were derived before 2001 and the cells are on the registry that NIH keeps that actually is made up of the cells that Mr. Bush approved at that time. But certain states have laws that, in fact, make it illegal, and I learned today that New Mexico may have such a law. So you need to sort of pay attention to where you live to figure it out. This may be unclear exactly whether it extends, but from what I understand, it does seem to extend to research that would involve human embryonic stem cells, because it seems to say that it would involve research or treatment, for that matter, involving any cell derived from human fetal material, and that would extend to stem cells. But I'm no lawyer, thank goodness. So it is legal by federal law, and but you have to use the approved lines, and it is legal in most states, although certain states may have laws against it. Is it illegal to derive new lines? No, it's not illegal to do that either. And again, the only prohibition is using federal funds to do it. And again, certain states may have laws. But new lines are being generated right now using non-federal funds. Private companies are doing it. We in California will do it with funds that are provided by Prop 71. So it is happening. Although it is happening at a rate that isn't up to full steam yet. As you know, Prop 71 is just getting started, and we had to go through some legal battles to actually get it going. And I can answer questions about that, but we have now actually sent out about $120 million in research, and we think we will have the legal issues resolved in the next couple of months and really be able to launch the program. So, is therapeutic cloning illegal? Actually, no, that isn't even illegal yet, although there are laws that are being proposed. And this is something that all of us need to watch. But any attempt to actually clone a human being is totally, completely, absolutely illegal. So that's where we stand right now. And just to quickly mention what California is doing, as you undoubtedly know, California voted in Prop 71. The purpose is to provide funding for the development of technologies involving human embryonic stem cells. Eventually, we can issue bonds totaling $300 million a year. We can do that over a 10-year period. So it is a total of about $3 billion. The bill created the California Institute for Regenerative Medicine, that's CIRM. Oversight is by this committee on which I serve. And just a point that is actually a little area of confusion. The money does not have to be spent in 10 years. The limitation is over the number of years that we can issue the bonds and how much we can issue per year. But once we generate those dollars, we can actually continue to spend it over a very long period of time. We intend to spend it quickly in order to try to get things going as rapidly as possible. But we also intend to spend it carefully to make sure that it's used in a way that is highly appropriate and that doesn't waste taxpayer money. Okay, and if you're interested in what's going on, the organization is extremely transparent in its activities. All of our meetings that actually make the decisions on how the money is spent are in public. We sit up on a stage. We on the committee are not allowed to talk to one another. It's against the law for us to talk to one another except in that public meeting. We can't even send emails back and forth and have emails that are called serial emails, where I may talk to my friend Sue Bryant, and Sue Bryant may then talk to Bob Klein or whatever. Those are also illegal. And we're actually very carefully monitored. There's a lawyer sitting with us at all times. So it's kind of amusing, actually, from the point of view of a working scientist to be in this kind of thing, but kind of fun. If you're interested in what's going on, www.cirm.ca, that's California, .gov. And boy, does that .gov hold sway over everything else. So you can log on to that and find out anything you'd like. Yeah, sure.
And then because of the unclarity on this bill in New Mexico, anybody that's come here, if they just bombard like the governor's office with an email or their legislature, that would be awesome, because then maybe we get more people.
I have enough trouble with California politics. I'm going to leave New Mexico politics to you. As I say, I'm here to tell you facts, and then what you do. I think you just did. Everybody hear that? Would you like a microphone? No. So I think that, I would just say that, I think that all of us need to find out what the law is in New Mexico. I do have an interest in New Mexico. My son lives here. He's a firefighter with Albuquerque Fire Department. So that's, so that's where things stand right now. In the next few minutes, I'd just like to sort of tell you where we are on the research. And what exactly is online and what the hope is. I think the hope is considerable, but there are, I guess, different horizons of hope. There are some things that are, I think, coming along relatively rapidly, and some things that will take time. So our biggest challenge is really twofold. The first is to be able to actually develop the strategies, and it really is it's technology. How do we teach stem cells to become particular types of cells? And so, there's a whole set of cells here: liver, neurons, hearts, and so forth. And we don't truly know how to do that very well yet. We've been able, talking now the field, have been able to cause stem cells to develop into neurons in general and certain of the other cell types of the nervous system, but about the best we've been able to do in developing individual types of neurons, say the neurons you'd need to treat Parkinson's disease that release dopamine, is about 80% purity. And that's not good enough, because that other 20% can develop into other cell types, including heart cells and liver cells and pancreas cells and teeth cells. And this is not the kind of thing that you want growing in your nervous system. That's called a teratoma. So understanding how to keep cells from doing that is probably the major challenge that we have in stem cell research. Making them do what you want, become a neuron, for example, keeping them from doing this other thing and growing into other cell types, which they're perfectly capable of doing. So where are we now? We really need to develop the techniques to cause stem cells to differentiate reliably into particular types of nerve cells. And as I said, at least in terms of nerve cells, our current techniques produce mixtures of cells that are about, that should be 25% impure. We're actually better than that. So what I want to focus on then for sort of the final piece is what I think is maybe a model of a short-term success, maybe. The one that I think is pretty far along toward a clinical trial, not clinical application, but clinical trial. So the idea is, let's ask the cells to do something that seems relatively simple. And by that I mean, if you're asking a nerve cell, even if you can make a nerve cell grow into the kind of thing that you need in the nervous system, you actually still need to get it to connect. Because, as we heard this morning, everything that our brain does is based on connections between cells. So you have to get these axons growing these very long distances. And even if you make them into a nerve cell, getting them to grow to the appropriate location is something that we haven't yet achieved. It's going to be tough. It's going to take a lot of different things. actually the closest I think we've come is an experiment, or rather report, that Doug Kerr just published a few months ago, where he did a variety of things to motor neurons to make them not only live in the spinal cord but actually grow out. But it takes a lot of steps, and so we're really not there to the point that we can do that reliably, although there's promise. So what we need to do is find something that's relatively simple for the cells to do. We need to have a definable target, a definable indication, something that's uncomplicated. We need to have predictable and measurable outcomes. And I'll just give you an example of that that is important for the diseases and disorders that affect the spinal cord, and in fact important for diseases that affect other parts of the nervous system. So you've already heard about this all day long, as a matter of fact. One of the things that happens after an injury, whether it be traumatic or something related to transverse myelitis or MS, is that axons die and the cells that provide the insulation on the wires die. And these cells in the brain are called oligodendrocytes. Okay? And they're actually a cell type that send out a really long membrane process that wraps around nerves. Imagine imagine a roll of tape, actually. And each of these cells is like a little roll of tape. And they unroll their tape and they kind of wrap that first piece around the nerve cell, and then they'll wrap and wrap and wrap and wrap and wrap. And it's those wraps, just like electrician's tape, that provides the insulation that allows nerves to work. So with a spinal cord injury, one of the things that happens is that some of these cells, these myelin-forming cells, now I'll use the word myelin because that's what it is. Some of these cells that make the myelin sheath die. And as a result, the axons are bare. So they short out, to use a simple analogy. They're alive, they connect, but they don't transmit in an efficient way. Now, how much demyelination there is, that is, how many of these axons are actually left after an injury or a disease, is not really entirely known. I don't think we have a good test of that yet. But it's becoming more and more clear that many diseases and disorders do have a component of demyelinated axons, and these then represent the targets for therapy. The idea is very simple. Take any cell type, but in this case we'll use human embryonic stem cells, cause them to become these myelin-forming oligodendrocytes, put them in the injured nervous system, and hopefully they'll make myelin. And they do. And in making myelin, they do cause at least some and an important degree of improvement of function. So I'll just take you through that. And let me just say that this is all the work of, I'm sorry it's too dark to see Dr. Kiersted's face down here. This is Hans Kiersted's work. He is an associate professor in our center, and he's been really a leader in the development of stem cell technologies. This paper that I'm going to just tell you about now was published a couple of years ago, and as you'll see, it's on the way to a clinical trial, and I'll try to give you a little bit of a timeline. So basically what his group did was to find a way to cause these undifferentiated stem cells. So this is actually a collection of them. There are probably close to 300 or 400 of these cells all in a ball. And he found culture conditions that made them turn into sort of the precursors of these oligodendrocytes. And just it happened that the cells that were the precursors turned out to be these yellow balls, and then there were the white balls in the culture. And he was actually able to isolate the yellow balls and then treat them in a series of manipulations to become these oligodendrocytes. So oligodendrocytes have these long processes, and this is that little piece of tape that's beginning to form here that if it was in the nervous system, it would go over and plant itself on an axon and form this myelin wrap that forms the insulation. So this is all in a dish. He was able to do this in a dish. So then what they did was to take these now, what we now call oligodendrocyte precursors. So these are cells that become oligodendrocytes. And he transplanted them into the nervous system. And so this is just sort of the preparation to that. And I guess the words on here are a little hard to read. I'm sorry. So the end product prior to this differentiation protocol is a set of cells that look like oligodendrocytes. They have these membrane sheets. They are expressing the markers that are characteristic of oligodendrocytes. And they're about 98% pure. But remember that there's a few other little troublesome cells over here that are not apparently, oligodendrocytes. And I'll come back to that in a minute. Therein lies our risk. But 98% is pretty good. So he took these cells, created an injury in the spinal cord that is a partial injury. So we who work on spinal cord injury try to use models that aren't too cruel to the animals. We all went into this field because we love animals. But we still need to study these devastating human diseases and disorders. And so we try to create models that we can see deficits similar to what is seen in a human, but that aren't so devastating that the animals are really tragically impaired. We create what we call a partial injury. It's actually a contusion injury, and it causes a small hole in the spinal cord. The animals are initially paralyzed in their hind limbs, but over a few days, they begin to be able to use their hind limbs and even to walk. So when you see these animals in the cage, they're healthy, they're able to eat, they're able to drink, they're able to get around. They actually are group housed because they are social animals. And really they look pretty much unimpaired, although when you take them out of the cage and if you put them down, you'll notice that they stumble. And I'll show you the way they look in a little bit. But you know, they have enough of a deficit that you can measure it and assess the effect of a treatment, but not so much of a deficit that they're really badly impaired. So what Hans did then was to, in this model, transplant human embryonic stem cells that were targeted to become oligodendrocytes, so they're called the oligodendroglial precursors, into a moderate contusion injury, and this was done 7 days after the injury. And he then tested these animals over long periods of time. And what he found was that the transplanted cells survived. They actually differentiated, so you can't see it here, and it doesn't matter. This is a stain for the cells, just showing you that they're there. We have markers for human cells. They actually migrate. So this is where we put them, and this is measuring at distances from where we put them. So they actually migrate and differentiate. So just what you want. They actually create myelin. So using a mouse that has a genetic defect that causes myelin not to develop at all, he was able to show that if he put these cells in, they would actually make myelin sheaths. So here you can see myelinated axons in a mouse that received human embryonic stem cells, and actually he was able to show that these came from the transplanted oligodendrocytes. So they survive, they differentiate, they make myelin. Great. What do they actually do for the rat? And I'll just show you a... so can we... Do you know how to start a movie in a Mac? If you move the hand over and touch, the movie should run. So I'm going to show you a video. If you show a single animal, you know, that animal may or may not sort of show the characteristic deficit. Should be right on the picture itself. There you go. Just touch that. Click on that. And it should run. There you go. So this is a movie, a cartoon, of a rat with a spinal cord injury that has not been treated. So he was initially paralyzed, and now he's recovered. And you can see that they stumble. So his leg isn't moving perfectly. He's able to walk. He's able to bring it under him, but it's not in the right position quite. Can you run it again? Just click it, yeah. And sometimes the leg slips out from under him. And we have a rating scale to actually quantify this. But you'll see he's not able to always get it under him, but he is able to bear weight, and you can actually watch them, walk around the cage. This is at slow motion, so you can actually see the feet moving. If the real rat was there, it'd be pretty hard for you who weren't used to watching rats to see what was going on. And then this is a rat that has been treated by receiving human embryonic stem cells. So if you could click that again, this should be the next video, I think. Yeah. So notice that the rat is able to get that rear foot under them. They're able to step, but the stepping is regular. The foot position is better. They don't slip and slide. They're not clumsy. And this is a situation that for a rat is really quite good. Now, I just want you to think with me here. The untreated rat is not completely paralyzed. He's able to walk, he's able to get around, but he's clumsy. The treated rat is better. Okay? But it's not something that goes from complete paralysis to completely normal motor function. We're not there yet, but it's a very, very, very important improvement in function for a rat, and I think probably for people as well. And this just is data that actually shows the improvements of untreated rats. So they're initially paralyzed. They receive the transplants at 7 days. This low level of function is the initial paralysis, and the arrow indicates the transplant. The rats that received control transplants, these were actually Schwann cells that were human Schwann cells, but not differentiated to become oligodendrocytes, improved to this level, so that was what I showed you. The animals that received human embryonic cells got that much better. So it's about a 3 to 4 point or so difference on this rating scale. That's a rat neurological rating scale. That's the good news. The bad news is that we did the same experiment on rats that had been injured for a year, actually 10 months out. And they didn't get any better. So this curve down here is actually measuring the rat's ability to walk on this same scale. Now, by now, these rats have been recovered for a long time, so they're at the level of these untreated rats up here. This is about as good as they get, so at about just a little under 12 on the scale. My pointer's running out here, but right about there. So, and then here at the arrow, one group got the transplants of oligodendrocytes. No change. Everybody continues at this level. So that's the bad news. Right now, we don't have a success at long periods after transplantation. So, where are we? Try to show you. Yeah. What are the issues? Well, I have to tell you that this is hard work. These are expensive experiments. And it is difficult to actually get the cells and grow them up under the conditions. Hans estimates that each experiment, just the cells, never mind the cost of the rats, cost of the technicians, everything else, takes about $50,000. Very expensive. So one of the issues is whether we need to have a complete replication of this work before we go on. I will say other labs have carried out similar experiments using different types of cells. Doug's lab is one, John McDonald's lab is one. These are, let's say, proof of concept experiments. Similar results when you transplant cells that make myelin, but it's not using these human embryonic stem cells. So we don't have a perfect, absolute replication. And, you know, when you're talking about moving things forward to clinical applications, there's a real issue of whether it needs to be replicated independently in somebody else's lab. And that hasn't been done yet, although the company who's been supporting the work and involved in generating the cells and in which I hold no stock, everybody hear that? I hold no stock. Geron has replicated experiments in-house, it is said, but they have not published that work. So one issue is whether the FDA will require an independent replication. We still don't know whether transplanted cells ever develop into tumors. Well, Hans and his colleagues have looked a year, but you know, if you're transplanting cells into humans, you're going to be living a long time after you receive your transplants, hopefully. So we don't know how long we need to go before the FDA is going to be satisfied that they don't show tumors. And rats only live about 2 years, so that raises the issue of whether we need to do these experiments in larger animals. And this is going to be the time lock. If the FDA requires lengthy, long survival intervals before they allow human trials, then we have to do them, and it just takes time. Timing, we don't know yet what the therapeutic window is. I showed you that 7 days works, 10 months doesn't. Every experiment is $50,000 or so just for the cells, plus all the rest. So how many time points can you do? Well, not very many on our bucks. And so this is an issue. We have a therapeutic window that seems to work, but you know, there might be a better one. We don't know. We don't know what the dose is. We don't know how many cells should be transplanted, especially for people. We kind of know this for rats, but it's going to be a guess in moving forward to human clinical trials of how many cells are actually required. And it's not yet clear whether experiments in large animals, especially primates, are going to be required before moving forward. So there's a lot to still be resolved. And the steps are completing the long-term safety testing, whatever the long-term is basically decided by the FDA, to see if these cells ever develop into tumors. Obtaining final FDA approval for a phase 1 trial. Having said all these caveats, Geron hopes, it's their projection, to begin a phase 1 safety trial sometime in 2008. And I just report that with no evaluation. Whatever the initial trials, at the basis of what we know right now, are likely going to involve transplanting cells 1 to 2 weeks after injury, because that seems to be the time frame that does work, and at least we know that much longer periods of time don't work. So we are there. One of the things that we also don't know yet in what proportion of patients demyelination is actually a significant cause of disability. I think that's going to be an important thing to sort out. And I personally think that we need to develop some sort of a test to be able to assess this in individual patients. And I'll just say, my own speculation is, I wonder if fampridine might work for this. For those of you who are neurologists out there, fampridine is the potassium channel blocker that went through a clinical trial and did not achieve a statistically significant benefit, but for many, fampridine is available on sort of a brown market, especially for people suffering from MS. Some people say it's great, some people say it isn't, and maybe the reason is that some people have significant demyelination and others don't. Maybe that could turn out to be a useful diagnostic tool. I don't know. I throw that out there for the neurologists to think about, but in any case, I don't think we have one now. Maybe Doug or John could comment on that later on. And finally, I just have to say that whatever we see at the end of the day, I think that it will be a legacy to these two wonderful people, Chris Reeve, of course, and Dana Reeve. They certainly were instrumental in bringing so many people to the bench, so many young scientists, getting people interested and excited about spinal cord injury research, and obviously in the last parts of their lives spent enormous amounts of time on the political side of things trying to make things happen. And so with that, I will leave you and be happy to answer any questions. Thank you.
Let me ask you